Good afternoon, everyone. I'm Stephen Burley. I'm the director of the RCSB Protein Data Bank, the Institute for Quantitative Biomedicine, and the RAD Collaboratory. I'm very pleased to welcome you to the last of our Wednesday noon seminars for the fall 2025 season. We're hosting today one of our own, Jay Serebrenik, from the RWJ Medical School. Before I introduce Jay, I'll just give a few housekeeping announcements and uh, I recognize the deadline for registration for Raise 26 may have passed, and I apologize for that if that's actually the case. There is my experience with uh, registrations these days is that they are almost always extended. So the RAD Collaboratory is pleased to promote Raise 26. This is an undertaking chaired by one of our colleagues, Dr. Jim Samuel from the Blaustein School. You can learn more about the opportunity to win $20,000 in prizes by going to the URL that's listed on the slide. There's also a 2D barcode if you prefer to use that. I want to reiterate that the International Union of Crystallography will be holding its triennial Congress and General Assembly in 2026 in Calgary, Alberta, Canada, August 11 through 18. There's still plenty of time for you to submit an abstract to give either a contributing oral presentation or a poster. The deadline being February the 15th. Registration for discounted rates ends on the 31st of March. There will be two RCSB Protein Data Bank workshops held immediately before the meeting on August the 10th. We'll be conducting a workshop with our Worldwide Protein Data Bank colleagues on PDB deposition and validation, and this will include deposition of macromolecular crystallography, 3D electron microscopy, and NMR structures to the PDB archive. And then on August the 11th, we're going to be focusing the workshop on integrative hybrid methods and specifically on the combination of 3D electron microscopy with chemical cross-linking to accommodate those structures as part of the Protein Data Bank archive. There's a variety of housing options available for delegates, ranging from expensive hotels to the local university dormitory, suitable for every budget and preference. The strength of the US dollar against the Canadian dollar makes this a very cost-effective meeting, and one that is only about 100 miles from the Canadian Rockies, which would be a terrific venue for a vacation either before or after the Congress. So I urge you to consider this possibility. So to the job at hand, it's a pleasure for me to be introducing Yevgeniy, known as Jay Serebrenik. He's an assistant professor in the Robert Wood Johnson Medical School and a member of the Department of Biochemistry and Molecular Biology. Prior to coming to Rutgers in 2024, he was a postdoctoral fellow at CHOP, the Children's Hospital of Philadelphia, with Ophir Shalom. And prior to that, he was a PhD student at Yale with Craig Crews. In talking with Jay before the seminar, I learned that he emigrated to the United States from Russia at about the age of 6 and was brought up in the Boston area and went to Tufts University studying biomedical engineering systems and graduated with a bachelor's summa cum laude. Jay's research interests are focused on understanding protein homeostasis across across the cell and developing novel proximity-based therapeutics that harness protein quality control effectors. So we're going to hear from him today about programming the proteome with pooled tagging. And I'm going to stop my share now and hand the podium over to Jay and go off mic, off video, and enjoy the seminar. Thank you again, Jay, for rounding out the seminar series.
Awesome. Let me just make sure this works. Okay. Okay. Thank you so much, Stephen, for the introduction and for the invitation. As Stephen mentioned, I'm a relatively new assistant professor at BMB in the School of Public Health, which is nearby. And today it's my pleasure to tell you about our work to program the proteome. And throughout this talk, I'll highlight some potential opportunities for collaboration that I think members of the IQB might find interesting, particularly whether they are trainees or PIs. So if you think that there might be some potential common interest that we could possibly work together, or if you know of other people that are working on similar topics, please feel free to reach out to me. So this is a famous simulation of a bacterial cytoplasm. And I like to show this because I think it illustrates very well the complexity of the proteome. Much of a cell's behavior, whether it grows or differentiates or responds to stress, depends on the composition and connectivity of the proteome. And this can be visualized and thought about as a biological circuit. Where many different inputs and outputs are all processed to control how a cell behaves. And much of the challenge of modern biology is to be able to decode this circuit, to understand, to be able to read out proteome states and understand how they encode cell states, and also to be able to write in new proteome states to be able to control cell behavior. So manipulating the proteome in comparison to manipulating the genome or the transcriptome is, in a large-scale fashion, is still quite difficult, but this is a problem that we're trying to solve in our lab. So today, I'll be telling you about our work and technology that enables us to read out proteome states, specifically in the form of protein abundance or localization patterns. And we're also able to systematically write in proteome states by harnessing endogenous cellular machinery that governs protein quality control or post-translational modifications. And this would allow us to manipulate specific nodes in our biological circuits or possibly shift the balance of the proteome altogether to then allow us to control cell fate and cell outcomes. So the technology that allows us to do this, that allows us to manipulate the proteome, is something that I worked on as a postdoc. It's called Spotlights, which stands for Scalable Pooled Targeting with a Ligandable Tag and Endogenous Sites. And the way I think about it in shorthand is that it's a way to allow us to manipulate the proteome in a pooled fashion akin to how CRISPR or RNAi allows us to manipulate the genome or the transcriptome. And the way Spotlight Cell Library works is that we tag many endogenous proteins throughout this library where each cell has one different endogenous protein tagged at any given time. And we tag these proteins with a domain that functions as a generic handle. And through this generic handle, we're able to perturb these tagged proteins in a pooled fashion. So we can tag these proteins with many different domains, but the one that we really like to use is halotag. And halotag is a covalent ligand binding domain, and it's really nice because there's a very wide range of very well developed commercially available ligands available for it. So we can use these ligands to perturb the proteome and measure the proteome in a wide variety of ways. We can use fluorescent ligands, for example, to measure protein levels and detailed morphologies and localization patterns. We can use halotag ligands that induce misfolding or degradation or changes in protein-protein interactions, and so on and so forth. So there's a lot of chemical biology opportunities to be able to manipulate our tagged proteins in a pooled fashion. And in the next slide, I will show you a confocal image of an actual spotlight cell library that is labeled with a fluorescent ligand, so you can see what that looks like. And this is a pool of HeLa cells, where again, every single cell has a different protein endogenously tagged with halotag and labeled with a fluorescent halotag ligand. And you can see in this image just the wide variety of structures that we're able to see. We have lots of examples of mitochondrial tagging, nuclear membrane, cytosolic bodies, nucleoli, Golgi, plasma membrane, cytoskeleton, nuclear bodies, and so on. So there's no apparent limit to the types of cell compartments that we're able to visualize and target with our approach. So the way I've organized this presentation, I'll first talk about the Spotlight's technology and how we can generate these pool tag libraries. I'll talk about how we've used this technology to read out proteome states and to write out proteome states. And then I'll finish off with some ongoing work in the lab. So pooled tagging originates from CRISPR screening technology. And you can think of it as creating a pool tag library is very similar to creating a CRISPR knockout library. There's just one small tweak in the protocol that turns what you can think of as a pooled CRISPR knockout library into a pooled CRISPR knock-in library, basically. So just like in CRISPR, we transduce cells with a viral library of expressing to express sgRNAs with one sgRNA expressed per cell. And then the one additional modification that we perform in this protocol is a transfection of these generic tagging reagents, which allows for our gene to be tagged. So zooming in on this step. So for tagging, we rely on homology-independent integration. And this is in contrast to other CRISPR-based approaches for endogenous gene tagging, like homology-directed repair, where those approaches are very difficult to scale because you need a lot of site-specific reagents. You'll need a site-specific guide, you need a repair template, and that's very difficult to create in a pooled format. With this approach, we simply need a site-specific guide, and the rest of the reagents that are used for tagging are completely generic and can be used for all the different sites. So our site-specific guide will create a double-strand break, and then our DNA cassette, which encodes the tag, which we linearize in some way, often by cutting it with its own dedicated pair of guides, will then integrate into the site of the double-strand break at some rate and get integrated into the locus through this homology-independent integration approach. So one kind of caveat with this repair approach is that it is prone to indels. So you may notice that for this reason, we target introns because they're much more tolerant to the formation of indels. However, our tag is surrounded by splice sites. So that enables our tag to get scarlessly integrated into the coding sequence, regardless of whether or not there are indels present in the gene. This strategy also allows us to add additional genes outside of the splice sites, like selection markers, and this allows us to use strategies like antibiotic selection to enrich for genomic edits and increase the proportion of tagged cells. So once we've performed the tagging, we label the cells with a fluorescent halotag ligand, and we enrich for stable, highly expressed fusion proteins by fluorescence activated cell sorting. And then this allows us to create a pure pool tag library. And then once we have this library, we're able to perform an experiment with a variety of different halotag ligands, and then analyze it by deep sequencing as you can with any CRISPR screen. So in our initial approach to test pool tagging, we wanted to tag proteins in a wide variety of compartments to be able to just visually see how well this technique is working. So we chose about 13 compartments throughout the cell, represented by 20 compartment-specific proteins. And we try to find tag sites within these proteins in a completely unbiased way. So we're targeting introns. On average, genes have about 9 introns. So we try to target all the introns in a given gene, and we designed 10 guides, up to 10 guides per intron, depending on the size of the intron. And in the end, for targeting about 265 genes, we created a guide library that was about 21,000 guides. And many of these fusions, they don't end up working because you can imagine just peppering a gene with a bunch of fusions without any prior knowledge, you're going to get disruption of protein structure, protein folding. But the termini are not the only sites in a protein that can, in theory, accept a tag. If you think about a protein in three-dimensional space, there's lots of surface exposed residues that could potentially accept a fusion as well. And these are the sites that we end up enriching for in our pool tagging approach. And in fact, once we generate our libraries and enrich for these stable, highly expressed fusion proteins with FACS and perform deep sequencing on these libraries, we see that tagging is actually quite successful. When we're looking at the gene level, so this is a little histogram showing the number of sites tagged per protein. At the gene level, we're able to target out of in our very first approach, almost 90% of the targeted genes. And on average, of the genes that are tagged, we have about 4 different fusion sites. So that allows us to potentially also compare against different fusion variations within the same library for the same gene. So we wanted to perform some experiments to verify that our approach is in fact enriching for viable protein fusions. And we did this in a couple of ways, one of which I'll talk about here. And we wanted to see whether tagging essential proteins was as easy as tagging non-essential proteins, with the idea being that if tagging is generally disruptive, it would be much more difficult to tag an essential protein because those cells would just die off quite quickly. So we took about our 260 genes that we targeted previously. We empirically figured out which ones were essential with a CRISPR knockout screen. And then in parallel, we tagged these genes with halotag as well, and compared the frequency of the guide abundance. So this is just looking at guide counts as a function of time in the knockout screen. And we see a lot of the guides, cells encoding guides targeting essential genes drop out of the pool quite quickly. And here is the results from the tagging data. And we split up sgRNAs based on whether they target essential genes or non-essential genes. And from this plot, we can see quite quickly that that we're able to recover guides against essential genes just as easily as we're able to recover guides against non-essential genes, which suggests that our enrichment approach does not seem to disrupt at least essential protein functions. So that was a quick overview of the Spotlight's methodology, and I'll right now have a brief aside to show you some ongoing work of how we're improving pool tagging. So Spotlight's, the original version, works quite well, and we were able to create many large-scale libraries this way. But we want to increase the fidelity and the utility of this approach. And right now we're working on a modified version for pool tagging called Spotlights 2.0. And this approach is is improves the tagging, the pool tagging methodology by giving us more control of the way proteins are tagged, which has a lot of very important implications. So the way we do this is through prime editing based protein tagging. No longer are we using homology independent integration, where donors, one of the caveats with Spotlights 1.0 is that our DNA cassettes, because there's no homology, they can get integrated either the expected way, but they can also be integrated inverted, they can be integrated in tandem. So even if we tag a site that is very well validated and we know that should produce a very good protein fusion, antibiotic selection for genomic edits will only enrich cells with highly expressed fusions up to a certain point, because a lot of cells will express the blastocytin gene, but the DNA cassette might be in the wrong orientation and splicing just might not occur. So by controlling the way the tag is integrated in the gene through a prime editing approach, we have a number of improvements to the technique. So one is that we would be able to do antibiotic-based selection that would post-selection produce fully purified libraries. So we would completely obviate the need for fluorescence activated cell sorting. And this would allow for creation of much higher quality libraries, as well as make this technique more widely accessible to labs, for example, that don't have access to FACS. With this controlled approach, we're also able to induce tagging in an expression independent way. And we are excited about this quality of this approach for tagging iPSCs that we plan to differentiate into neurons, and specifically tag genes that are expressed exclusively in neurons but not in iPSCs. And this is something we would not be able to do with the existing approach of pool tagging. And another exciting advantage of our Spotlights version 2.0 is that we also have control about whether the tagging is heterozygous or homozygous. So because the tags all get integrated uniformly, one tag per site, we can combine donors with multiple selection markers and specifically select for cells where we have integration of both donors, which would indicate we have homozygous tagging, or conversely, we could specifically enrich for heterozygous tags as well. And so to give you a brief look into how this work is progressing, here I'm showing the work of talented Dr. Kathy Larimore in our lab. And so this is flow cytometry data of cells that are in the process of being tagged at a specific site in beta-actin. And on the x-axis, you see autofluorescence. On the y-axis, you see the signal of the tag. The far left chart is showing the untagged parental cells where we don't see any tagging. And in the middle chart, we see beta-actin tagged with our original pool tagging approach. And after selecting for about 3 weeks, we see an enrichment of about we see about 40% of the population expressing the actin fusion. And these cells that are not expressing the actin fusion, they have the genetic the edit, but it's just the donor, the cassette is probably inverted, put in the wrong orientation that doesn't allow for proper splicing to occur. With our new approach, after selection, in our first attempt without any additional optimization, we're able to get to 85% of cells that are positive for tagging just after selection without any kind of sorting. And you can see that the population is a lot more uniform. We don't see this additional secondary population, which might correspond to situations where cells are tagged twice in tandem So in those cases, actin might have two copies of halo tag fused to it. And we can see by fluorescent imaging that we get the expected actin localization pattern. So we're very excited and further developing this approach, expanding it to more sites and further optimizing so we can get fully uniform purified libraries after antibiotic selection, which would allow for even more different types of pool tagging related experiments than we can even do now. Okay, so how can we use our libraries once we create them? How can we use them to read and write out and manipulate proteome states? So the picture that I showed you before, where we see a mixed population of cells in a fluorescent image, that's a very powerful approach that allows us to look at all of the cells in our pool and analyze all the different localization patterns that we see. But the problem is, how do we know what protein is tagged in each of these cells? And how do we know not only what protein, but what site in that protein is tagged? So there's a very clever technology that allows us to figure out just that, which is called in situ sequencing. And in situ sequencing allows us to simultaneously image our pooled population of cells on a fluorescent microscope and subsequently perform sequencing of the sgRNA barcodes that's in each cell directly on the cover slip. So the way this works is that we plate our cells in our dish. We perform high magnification phenotyping of the different proteome patterns, protein patterns that we see. And then after we do that, we take literally an image of every field of view in the plate. Then we permeabilize the cells and then amplify the sgRNA sequence that's in each cell using a technique called rolling circle amplification, where we take the transcript that has the sgRNA sequence from reverse transcription and then rolling circle amplification that basically creates a big ball of that is enriched for this barcode in each of these cells. And then we can wash these cells with fluorescent nucleotides that'll label one base at a time. So we wash them, we image, again, every single field of view on the plate, and that'll give us information for the first base in the barcode. Then we cleave off the fluorescent group, we wash with another set of fluorescent nucleotides, and we take another image. And we just do this sequentially, imaging and then washing with fluorescent nucleotides until we're able to see on the cells directly what is the sequence of the barcode that is in the cell. So this approach allows us then to take an image like this, where it's a mixed population of many different cells, and turn it into single cell albums, where we know for each cell not only what protein is tagged, but exactly where it is tagged. So here are some examples throughout different compartments. Here's calreticulin tagged at intron 1 versus intron 7. So one is more N-terminal, one is more C-terminal. Calnexin, we see mitochondrial proteins, nuclear proteins. And so we were able to then start to look at individual tags and see exactly how these tagged proteins behave. And this actually provides us another opportunity to assess how well tagging works, because now we can do something very simple, just look to see whether the localization patterns that we see in our imaging match the expected localization patterns. So here we're looking at different guides, different tags, categorized on the x-axis by what compartment they're targeting, and the color corresponds to what localization pattern we actually see. So if it's gray, we have the expected localization pattern. If it's some other color, it has an unexpected localization pattern. So for the most part, we see that tagging does lead to expected localization patterns for the validated proteins, but there are some exceptions. But the nice thing about pooled tagging libraries is that they're completely iterative. For subsequent libraries, we don't have to, we can filter out the guides that don't work and selectively focus on the ones that do, that work robustly. And so our initial, what I showed you, our initial library that targeted 260 genes with 20,000 guides, that was a completely unbiased approach. But now that we know what guides work, we can drastically reduce the size of the guide library to create the same tag libraries. And so initially, all this analysis was done manually, which took multiple people a very long time. But we have since also figured out how to do automated image analysis, where we create our pool tag libraries, label them with a fluorescent ligand, do in situ sequencing, create these albums, and then use vision models. Like in this case, we use the variational autoencoder, which is based on a convolutional neural net architecture. That is a self-supervised model that allows it to learn basically to quantify these different localization patterns and embed this information in a latent space. Then we can take these latent spaces, which are very large, and perform dimensionality reduction and get a bird's eye view of the different localization patterns. So every single point here in this chart is a different cell. And from this latent space parameters, we have these cells cluster in specific groups that are very robustly specifically associated with certain localization patterns. So if you just pick out, we know the identity, we know what pictures are associated with all of these cells. So if we just pick out a cell from any of these clusters, we see that they have exactly the expected localization pattern. So from analysis like these, we're interested in seeing what is the proteome composition of the cells that we're studying? How do they change in different perturbations, whether they're genetic or pharmacological? Can we see global shifts in the proteome where specific proteins change? And then can we narrow in and study specific proteins that might be responsible for whatever the perturbation is that we're doing? So this allows us to read out the proteome states pretty quickly. So summarize this part of the talk. I've told you about our spotlights tagging method, that we're able to tag endogenous proteins in a very large scale way. And this tagging is high fidelity and comprehensive, and is enriched for functional fusions. And we can use a variety of image analysis techniques to be able to map these proteomic states, and ultimately we hope to be able to study how the proteome changes under different conditions. So this brings me to my first kind of collaborative challenge that I kind of open up to IQB, where we are interested in. So I told you about our variational autoencoder approach, but we're interested in developing next generation vision models to be able to perform similar tasks, but with higher fidelity, and to be able to integrate all of the information from our imaging approaches. So specifically, the previous approach, we weren't able to feed different channels into it, for example. So, but if we have a self-supervised attention-based model that can incorporate every single channel in our confocal images, so we can have a channel not just specifying the tag, but also channels for the nucleus, for the mitochondria, for the Golgi, for the ER, and seeing how these tag patterns are related to these other structures in the cells. That would give us a lot more information on the diversity and specificity of these localization maps. So we're interested in building these advanced models, using them to chart the proteome in detail, and ultimately we're interested in applying them to cells as complex as neurons that have which are very large cells, which have long projections that span multiple fields of view, And one of the challenges we're trying to solve is to be able to combine this analysis of tag localization together with, well, how do you reconstruct a full neuron in a dish and be able to study localization patterns, not just in the soma, but in the projections as well. And another challenge that we're interested in is trying to understand, how does protein tagging work? And can we basically understand what sites lead to highly functional protein fusions and which ones do not? So we have existing data that we're mining for this, but we also have a lot of exciting in development data. And so I think it would be amazing to be able to computationally be able to predict high quality fusion sites across all proteins and understand why some sites tag well and some others don't. And this will also be very useful in the design of proteome-wide pool tag libraries, where we can, rather than sampling sites in an unbiased way, we would be able to know exactly what sites to go for and have focused libraries or libraries with a constrained size that's still able to capture the breadth of the proteome. Okay, and now I'll talk briefly about how do we use pool tagging to manipulate the proteome. So we can visualize it with the fluorescent ligands, but we have all these other halotag ligands that can manipulate the tag proteins in different ways, all in a pool format. So there's different strategies that we have. One is to use halotag ligands that can induce some kind of perturbation, whether it's degradation or in this example is misfolding. So we were able to use a library where we had halotag localized to various compartments. We used a particular type of halotag ligand that induces halotag misfolding. And we used this technique to study spatially restricted protein quality control responses in different compartments of the cell. And I won't talk any more about that, but you can read more about that if you're interested in that work in the reference down below. We can also use these libraries to induce protein-protein interactions, and that will help us to identify proteins that we can harness to be able to modify protein states in a more precise way. And I will focus a little bit more on this work. So basically what I'm presenting is a large-scale, high-throughput induced proximity screen. So induced proximity is a very exciting field that has really kind of exploded in a little more than the last decade. But the way this works is that we're able to harness the cell's endogenous machinery, known as an effector, to act on a target of interest in a particular way using some pharmacological or genetic approach. And in theory, we should be able to harness any aspect of cell biology to be able to modify our target. We should be able to degrade these our targets, stabilize them, disaggregate them, relocalize them, change post-translational modifications. And probably the most well-developed proximity-based effect, I would say, is targeted protein degradation, where you use a bifunctional ligand to recruit an effector like this VHL E3 ligase as an example that ubiquitinates the target and leads to its degradation. And this approach has been very successful, both in basic research and in translational research. But it's only just scratching the surface because for clinical trials, for example, last time I checked, there was only about 2 different effectors that are used. And in basic research, there's just a few handfuls more. Meanwhile, there's hundreds and hundreds of additional proteins that are part of protein quality control that we could similarly harness to be able to modify our target in a particular way for degradation, but also beyond. And why might it be interesting to have more than two effectors in your toolbox? Well, that would be for numerous reasons. Chief among them, that different effectors might have different substrate preferences. They might have different tissue expression patterns. They might have different essentiality profiles, which might be important for translational work, where you're trying to decide for whether a cell might develop resistances to drugs using these effectors. So we're interested in using our approach to explore effectors in a systematic way and to expand the toolbox of effectors used not just for degradation, where there are already very few effectors, but for all these other kinds of protein effects as well, for which there are, for the most part, very few to no effectors available. So the way we approach this is using our pooled tag library, where we tag endogenous proteins with a ligand binding domain and use generic bifunctional ligands to recruit our tag proteins to a specific target. And in our initial development of this method, we use targeted protein degradation as a readout just because it's the most well-developed effect. So for this pooled tag library, we targeted members of the protein quality control network. We had about 760 members of the ubiquitin proteome system, autophagy lysosome pathways, chaperones. Here's some images of the library, where these are our pooled tags. And one of the targets that we used is mClover3, which is similar to GFP, which is a green fluorescent protein that is tagged within orthogonal ligand binding domain FKBP. So we can design screens to enrich for a particular effect in a lot of different ways. Just like you can adapt CRISPR screens to any kind of readout, you can adapt a pooled tag screen to any kind of readout as well. So for looking for effectors of degradation, we used two different approaches. One was a fluorescence sorting-based approach, and one was a growth-based approach. And they used different targets, but they used the same library. So we took our library for one of the targets, we looked at fluorescence of the cells that all were expressing mClover3 fused to FKBP. So for this, for the sorting-based approach, cells that that had reduced fluorescence intensity, we were able to sort those out and then do bulk sequencing and identify what tags were associated with that subpopulation of cells. We also had an endogenous target, endogenously expressed target, BRD4. And BRD4 degradation is known to be quite toxic. So this was a simple growth-based screen where we recruited our effectors to BRD4 with a ligand that bound to both endogenous BRD4 and halo tag. And we cultured these cells for a while, and the cells that led to BRD4 degradation dropped out of the population. So after a couple of weeks of culturing, we performed sequencing on the library and figured out which tags dropped out of the population. So these are the results from the screen. These are two MA plots. On the left, this is the sorting-based screen. This is the growth-based screen. On the x-axis, we see counts. And so every point here is a tag, different tag targeting a particular protein. So we see counts of the tag on the x-axis and the fold change on the y-axis. This is just a sample. I labeled some of these hits, and a lot of these proteins that we see act as viable effectors of degradation, proteins that have been established to work already in targeted protein degradation, including cereblon and other proteins as well. But besides these already previously known effectors, we discovered a lot of additional ones that we were quite excited about, including other E3 ligases, members of the CTLH E3 ligase complex. We saw hits that were parts of autophagy and even proteins without clear proteostasis roles and how they're functioning to induce protein degradation is something that we're interested in learning. So just I'll show you very briefly, just a little bit more in depth on some of these new effectors, including I'll focus on WDR26 and RAMBP9. This is flow cytometry-based analysis of degradation. Blue, we have vehicle control. Green is a positive control where we're looking at degradation of FKBP-fused GFP. And DTAG V1 is a small molecule that binds to both FKBP and the VHL-E3 ligase. So it's a pretty potent effector of degradation. And WDR6 we see is able to degrade cells quite well as DTAG V1, and in some cases even better. And RAMBP9, which is another member of this complex, also works quite well. You have to remember this is also log scale. So we still see, so even though it doesn't work quite as well as WDR26, it still works quite well. And we can do dose response curves. We see this characteristic hook effect, which is often seen for drugs that work in this way, like Protacs. And in time courses, we see that with increasing time, we get increased degradation with WDR26. Leading to almost complete degradation in under a day. And similarly, we can look at our BRD4 target. Here is our by Western, here's our BRD4 row. And we see when we recruit WDR26 at a variety of different concentrations, BRD4 gets completely wiped out. Meanwhile, if we recruit an inactive effector or a parental cell line, there's no effect. So to summarize, we are able to use our pool tag libraries to do systematic protein-protein interactions. And we're able to measure them in a wide variety of ways, which is conducive to looking for different types of effects. And we're interested in using these recruitment-based screens to identify protein function in an unbiased way without basically, it's a type of functional proteomics that does not rely on gene knockouts. So can we just bring a protein to another protein and see what happens? And we want to use this approach to identify proteins that can do not just degradation, but a wide variety of different functions as well. And so that brings me to my ongoing work. So one of our projects is to look for modifiers of protein aggregation through an induced proximity-based approach. So for all these approaches, we need a good model. And so for our aggregation experiments, we have been so far using Huntington. And Huntington is the chief pathogenic protein in Huntington's disease, which has a CAG expansion repeat. And when this is expressed and processed, it leads to these very well-characterized inclusions. And so we are asking simply, well, can we find a protein that when we bring it over to these inclusions can affect the inclusion in some way? So to make Huntington compatible with our recruitment system, we took mutant Huntington exon 1, used it to FKBP, this orthogonal ligand binding domain, and GFP for visualization. And so this is work of a talented graduate student in my lab, Rahul. And so here Rahul is showing that we can visualize our Huntington model in two very powerful ways. One is through confocal microscopy, where we can see that upon induction of expression, we get the formation of these these uniform, discrete inclusion bodies, despite the fact that we have all these additional domains attached to Huntington. And we can also track the expression of this protein over time by flow cytometry. And there's also certain parameters in flow cytometry data that we can use to also measure the percentage of cells that have aggregates or not. We've also confirmed that this model is compatible with recruitment through a couple of different approaches. Shown here is through a FRET-based approach where we overexpress in our Huntington lines halotag fused to mRuby3, which is a red fluorescent protein and is a FRET acceptor for mClover3. And flow cytometry analysis shows us that with the recruitment-based ligand, we see an increase in the FRET signal, suggesting that we are getting recruitment of mRuby3 to mClover3. We can also show this in a microscopy-based approach, where in the DMSO condition up top, we see mRuby3 halotag localized throughout the cytoplasm, but then with the addition of the ligand, it gets pretty tightly localized to the Huntington inclusion. And this is just a single cell we're seeing here, but we've also done large-scale quantitative image analysis where we image thousands and thousands of cells, do cell segmentation, puncta segmentation, and then analyze each cell individually. And we've calculated an enrichment score for a given fluorescent channel to see how much of a signal enrichment there is in the inclusion. And when we're looking at mRuby, we see that in DMSO, there's very little enrichment, but with the introduction of the ligand, we see a pretty big increase, and every single point here is an individual cell. And we see that almost every single cell responds to this ligand. So we're currently building our pool tag libraries to allow us to do these recruitment screens. The nice thing, because this is a pharmacological based protein-protein interaction, we can do variations of these screens. I think that can give us a lot of interesting information. For example, we can induce recruitment after we induce Huntington expression or before we induce its expression. And that might allow us to differentiate between disaggregases or just inhibitors of aggregation. So this is ongoing work, so stay tuned for more from this project. And then one additional project that I want to highlight is our modifiers of protein folding. So our model for this project is p53, which is a very famous tumor suppressor transcription factor, which normally binds to p53 motifs and induces expression of genes associated with cell cycle arrest or apoptosis. However, p53 mutations can often lead to destabilization of the protein, which can give it gain-of-function effects, lead it to bind other targets, and can sometimes lead it to act like an oncogene. So our experimental design is to see whether we can find a protein that, when recruited to p53, can possibly refold it and reactivate it and restore the tumor suppressor activity. So here I'm highlighting the work of Dom Spera in my lab, and he also developed a similar model compatible with our recruitment approach, where we took a mutant p53, fused it to FKBP and mClover3. Imaging shows that it has expected localization patterns. The wild type is exclusively nuclear. The mutants that we're interested in have a little bit of a less of a nuclear localization pattern. And when testing it with a transcriptional reporter, we see that the mutants compared to the wild type have drastically reduced transcriptional activity. So using this as the basis for enrichment, we are planning on performing a recruitment screen where we can use a transcriptional reporter to enrich for cells where p53 activity has been restored, either through a fluorescence-based sorting approach or, like demonstrated here, potentially through expression of a cell surface marker, which we could use to rapidly enrich for cells with strong p53-related expression. We're also interested in using localization as a readout and possibly cell growth as well. So this also brings me to the third collaborative challenge that I want to pose. And for the effectors that we find, we're interested in being able to manipulate them without necessarily tagging them, so that we can explore them in other cell types where that haven't been tagged or maybe are not amenable to tagging. So we're interested in designing short proteins that can bind these effectors or bind the targets and use these to develop genetically encoded inducers of proximity so that we can use these tools to manipulate protein states to however we desire. And in a similar challenge, I think it's also interesting to know how does the 3D geometry relate to activity when you induce protein interactions. So can we mine our existing protein-protein interaction data to find active surfaces and identify which parts of the protein need to interact for us to see an effect? And this is an example from our previous work where we took this protein that I described earlier, WDR26, that was fused with halotag. And this is an alpha fold-based representation of this fusion model. And it kind of gives us an idea of how this fusion might be working. So WDR26 has this beta propeller domain, and halotag is shown here kind of floating adjacent to this domain with this ligand cavity pointing directly out over it. So what we think is happening is when we're recruiting a target to halotag, it's positioning it right above this beta propeller domain. And that's why we think this effector fusion is as potent as it is. So I think there's more to discover from these types of studies. All right, so I'll end with this. I'll summarize that our pooled protein tagging manipulation approach has been very useful to allow us to read out proteome states and to allow us to manipulate proteins to discover effectors that can modify proteins in very particular ways that we can then harness to perturb the proteomic network in particular ways and possibly control cell outcomes in both basic and translational approaches. So I want to thank my wonderful lab. Pictured here, I'm very lucky to be working with all of them, as well as our funding from Robert Wood Johnson, Rutgers, and the NIH. So I'll take any questions.
Any questions, or the chat and Q&A online should be open now if anybody watching has any questions. Bueller?
So I see no, thank you, Jay. That was very impressive. I see no questions in the Q&A section. I'm just checking the chat now.
We got Greg with a question.
Greg with a question. Thank you very much, Greg. Please proceed.
Okay. Two questions. The first is a very quick one, just to make sure I'm following correctly. Does CAG code for glutamine? You mentioned
yes, polyglutamine.
Okay.
Yes.
And the other question, in terms of the effectors you're looking at, particularly those last two models you're referring to, has you or anyone else looked at HSP90, for example, and similar proteins as effectors for some of those systems?
We're definitely making sure to include these in our screens. But as far as I'm aware, there's been no studies where people have specifically recruited HSP90 to these aggregates to see what the effect is. People have used PROTACs and effectors of degradation to degrade inclusions in several cell model systems. But none of these tools, as far as I'm aware, have been able to disaggregate or dissolve these proteins.
Thank you.
There is a question in the chat from Arthur Zalewski on the RCSB PDB team at UCSF, and he asks, is it possible to do double tagging?
Yes. Yes. So Yeah, there's no reason we can't do multiple or orthogonal labeling. Um, and we are interested in approaches where we further tweak the system to enable us to do, interesting combinatorial based interactions. So yeah, this is a direction we're interested in pursuing.
Jay, I'd like to take the chair's prerogative and just ask the last question, please. Uh, I'm interested. This is more general. I'm interested in your thoughts about why it is that degraders have not seen as much success in the clinic as people had hoped for. It's been an area of intense interest, but in terms of the number of US FDA approvals, it's I would say it's been disappointing for some people at least, particularly venture capital investors in degrader-based companies.
I think well, for one, I think it's still early. I think a lot of these, clinical trials are relatively recent and a lot of them do have promising data. But I think why maybe they're not more, home runs is because we are only working with a very limited number of effectors. And for example, both cereblon and VHL, which are primary two effectors used in clinical trials, particularly for oncology purposes, the cells have a lot of mechanisms to downregulate them and to develop resistance against drugs that harness these effectors. So having a much larger effector toolbox, I think, will be able to we'll be able to exploit so much more biology and make these recruitment-based drugs so much more robust. And these WDR CTLH-based effectors that we discovered, for example, these ones are known to be highly expressed in a lot of different types of tumor tissues. So, and there's also evidence that some of them might function as cancer drivers. So these might be interesting candidates to try in comparison to cereblon and VHL because those types of effectors might be much more difficult for a cell to downregulate. And if they do, maybe then they'll just die. So I think there's still a lot more work to be done in this field.
I concur, and I just want one related question. One of the reasons that some people don't like these degraders is that they say that the small molecules are not going to be very drug-like and are going to be hard to deliver.
Yes.
What are your thoughts on that area?
I think this is also an area in active development, and there are plenty of exceptions to this rule. And there have been, I think, PROTACs reported that can even cross the blood-brain barrier, I believe. So I think there is development that can be done to further optimize this. And there's other types of drugs that can induce proximity. There's also molecular glues, which have more drug-like properties, which they work less like building blocks like a PROTAC, but they basically function the exact same way. And potentially using genetically encoded effectors could alleviate or could address the problem from a different angle as well. So I think here there's also a lot of things to be developed and explored, and I don't see why this, several years down the line we won't see robust drugs that work for different indications.
Thank you. So let's please all join me in thanking Jay for a great seminar and a terrific close to our very stimulating fall 2025 series. Thank you, Jay.